Hi everyone, and welcome to the next webinar in our new Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you're having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all the associated materials within a few days. If you have questions during today's webinar, please let us know using the GoToWebinar chat box. Today's talks will be 20 minutes each, followed by 10 minutes for questions. Benjamin Sweshinger, who is a future fellow and independent group leader at Australian National University, is with us today to moderate the questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars, and you can learn more about ASPB, the opportunities we provide, at aspb.org. This webinar is the second in our new virtual talk series, which was initiated thanks to the efforts of Benjamin Sweshinger and Jürgen Kleinven as a response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and sign up if you'd like to be considered for a speaker, or if you would like to donate or join as a sponsor. You can find us on social media at plantae.org and at ASPB. For those of you listening to this as a recording, feel free to reach out to us on Twitter with follow-up questions or comments using the hashtag PlantaePresents. Thanks again for joining us today. I'm now going to turn it over to Benjamin to introduce our speakers and moderate today's session.
Thank you, Katie. I actually wanted to say a couple of words too before we go over to Dominique. So yes, I would just like to echo what Katie just said. If you like this, it's a great opportunity to sign up for ASPB as your plant science society. Also, we got a lot of different people who want to sign up for talks. So please give us a little bit of time working through these many different suggestions. At the same time, we also are aware that other people organizing seminar series too in the plant science space. So the global calendar is a new place, like a place on the Plantae webpage where you can find those, and we hope to collate them in future. Of course, this is all only made possible because we have support of ASPB. So if you're interested in supporting this, your sponsorships, get in touch, as Katie mentioned. And if you are interested in helping out, shaping this seminar series in future, please get in touch with us too, because we have so many speakers, people or potential speakers, we are exploring new opportunities and ideas of how to make this happen. And last but not least, I would like to put my thanks to Mary, Katie, and everybody else at ASPB who really pulls us off in a very short time. All right. That's all for me. So with no further ado, I would like to introduce Dominique Bergman. She's a professor at Stanford University. She was an undergrad at the University of California, Berkeley, and received her PhD from the University of Colorado Boulder, followed by a postdoc at the Carnegie Institute, Department of Plant Biology. Her work focuses on specialized structures called stomata and the role of asymmetric cell division and cell-to-cell communication in cell formation. I'm really excited about the talk, and I hand it over to Dominique Bergman. Thank you.
Okay, thank you very much, Benjamin. And I want to really also echo my thanks for Benjamin and Jürgen and Katie for putting this together. So I chose a title which I think is appropriate for what we're all facing, which is adjusting to a changing world, but with an emphasis on stomata and emphasis on plants. So I need to figure out how this works. My webcam, as part of the pandemic, also died. And so I will be presenting without you seeing me, but this is a photograph of me with some information. I'm at Stanford University. I'm an HHMI investigator. And for the questions that might come after this, you can reach me at Twitter. And again, using that great hashtag, Plantae Presents. So I think we're all quite aware right now that the collective activities of many, many individuals are affecting the global climate. And immediately that means what people are doing now that was different from a month ago or several months ago, and how we're already seeing the effect on the climate. So for example, this is something that many of you have probably seen, which is satellite images over China in the beginning of the year of 2020, and then a few weeks later, where after the pandemic was causing changes in behaviors of people, and there wasn't more traveling, and you can see NO2 in the atmosphere has dropped precipitously. This is one example. An example that's closer to home to me in California are measurements that are being done on carbon dioxide. So in this graph, which hopefully you can see my cursor for, this is following what CO2 levels are doing in the Bay Area. And when before we were in this COVID-19 situation, they would fluctuate daily, but they were peaking around 490 ppm. But after several different types of changes, people started working from home, and then more and more shutdowns were happening. We see that trend line is dropping. And so the collective activities of many individuals really do affect the global climate. Now, on a more positive note, perhaps, the collective activities of many individuals, meaning many cells on many plants, can also affect the global climate. And so my focus today is going to be on this pair of cells, the stomatal guard cells, and their role in affecting many aspects of the plant and many aspects of the globe. So stomata are epidermal pores. They open and close using a turgor-driven mechanism that we'll probably hear about a bit more from the next talk. And they can allow carbon dioxide from the atmosphere into the plant to be used for photosynthesis. And then they also are transpiring, so water and oxygen are leaving the plant, water coming from the roots, soil, all the way through the plant and into the atmosphere. And so the activity of these stomata is absolutely essential for plants, for them to be able to provide food for themselves and also to get water through them. But the activity of these has had an influence on the climate for many millions of years. So in a set of images that I have on the corner, we have plants of different ages. So here's a fossil of Cooksonia. It's about 400 million years old. These plants had stomata, and it's thought that these early land plants are actually were responsible for much of the atmospheric content that we have now. As we stand now, plants of many different varieties, some of them in natural ecosystems, some very large, some very small, and some more in cropping systems, they have influences on the climate. So you might notice that there's also an arrow going the other direction, and that is to say that plants in many ways also respond to the climate. So they're influencing it, and that influence is quite dramatic. For example, it's estimated that the entire water content of the atmosphere is recycled through stomata twice a year, and about 30% of the atmospheric carbon dioxide enters the biosphere through stomata. But plants are also sensing those things about the environment and changing their behavior. So that's famously known in terms of opening and closing of stomata. But there are also other elements, and one of those elements has to do with development. So opening and closing can happen over the time scale of seconds, minutes, hours, but development takes place over days, weeks, months. And these kind of developmental responses, in some ways, can be very helpful in adapting the plant to the environment, but they can also hard lock them into it. So what I have over here is a set of images of a coffee leaf, so a very important plant. Coffee grown in the shade versus the sun has very different numbers of stomata and other epidermal cells. So these developmental responses are really important for creating plants that are well suited to their environment. Now, there's a kind of, I'm going to define the kind of developmental response that I'll refer to during this talk, and that is something that's plastic during the life of the plant. There are developmental innovations. For example, in grasses, guard cells are not just two cells like this, but they've got two squished together cells and two subsidiary cells, and that whole unit is much more responsive. Michael Reissig in Heidelberg is working on trying to figure out how these cells are made. But for now, I'll talk about guard cells and the creation of more or fewer on a leaf. So back to Arabidopsis, which is a plant that we do a lot of our work in. Something as simple as changing the light can triple the number of stomata depending on conditions. This is work from Graham Dow. And this really fascinated us. It fascinated us because how is it possible that you make such a, you have to make stomata, they're essential. You have to make the other cells in the epidermis, but how do you change the numbers of them? And it turns out that work that was done a number of years ago really traced out the pattern of development. And to me, this is really exciting because it allows us to ask general questions about development in an environmentally responsive tissue. So to make stomata, to make this endpoint here, this purple pair of guard cells, there are a number of cells that enter this lineage. This is a cartoon of the events that happen. So these cells divide asymmetrically. They go through a stem cell-like phase in which they can divide many, many more times and amplify the lineage. And then they make choices to go down one path or another path to become one or the other type of cell. We call this an initiation phase, an amplification phase that has lots of asymmetric cell divisions, divisions that are different sizes and different identities, and then a differentiation phase. So the kinds of questions we can ask are about how do you make specialized cells? How do you make guard cells? How do you make those cells in distinct patterns? It's important that guard cells are not right next to each other, otherwise their opening and closing is hampered. And how do you tune those to the environment? How do you make those in the appropriate amounts? So I showed you a cartoon, and cartoons are nice, but the real thing is nicer. So this is an image of a young Arabidopsis leaf. Every cell is outlined by a plasma membrane marker in red. And then what you see are some green cells and some blue cells. And those green cells are the stem cells that I had in the cartoon. And the blue cells are ones that are committed to becoming stomatal guard cells. So what we can do is create movies in which we're watching these leaves develop in real time. This is about a 24-hour movie. And what we'll see is that all over the place, those stem cells are dividing over and over again. We're making lots of green cells. The leaf is growing, and about 24 hours later, we have a lot more blue cells. So a lot of cells that are now becoming those stomata. We can zoom in a little bit and see these events a little bit more clearly. And so in this movie that will loop around a few times, you can see a number of things. So you'll see what looks like movement, that's actually growth, because plant cells don't move during development or ever. They're locked by cell walls. So what you're seeing is this set of cells growing. Right in the middle of the image is a mature guard cell already. This is a pair of guard cells. And all around it, we see cells at different stages. So this is a very interesting one to watch. Here, we've got a stem cell, one that will divide asymmetrically. For a moment, we have two cells that are potentially stem cells, but they actually communicate with one another. And as a result of that communication, they change their identity. So one turns blue and will become a stomatal precursor. The other will actually take a different path and become a different type of cell. So back to this cartoon. We're interested in these various steps, and then the steps that have some ability to be changed by the environment. Now, I can describe what this lineage is, but work from a number of labs over the last 2 decades or so has really defined the key molecular players. So at the heart of this lineage are a set of transcription factors. They're called speechless and mute and FAMA, and they're major regulators of these different stages. I've color-coded them here so you can see where they're expressed and also where they're active. When you were looking at that movie, the green was speechless, the blue was mute. And these are transcription factors. They don't work alone. They have partners. They're called SCREAM1 and SCREAM2. They're obligate heterodimer partners. That's lovely work from Keiko Tori's lab. And these are major regulators. So in the absence of them, this lineage gets blocked. So in the absence of speechless, there are no asymmetric divisions. There's no initiation of the lineage. All of the cells are these lobed pavement cells. There are no stomata at all. Now these plants will make a few leaves if you feed them some sucrose, but they really don't do very well, and they'll die pretty soon after that. So it's absolutely essential that you have this lineage. Now, these are powerful transcription factors. If you lose them, you don't get stomata, but if you overexpress them, make too much, then you can essentially make all of the cells into stomata. So for example, if you overexpress mute here, all of the cells will turn into stomata. So speechless, mute, and FAMA are BHLH type transcription factors. This is a very big family in plants. Speechless, mute, and FAMA happen to be in one subgroup, and that's turned out to be very convenient. They've got a number of features that allow us to find those across many different plant lineages, and that's been really powerful. Okay, so these are, I'd say, at the core of creating stomata, but they're regulated. There's communication among neighbors. You saw that in the movie where the two green cells didn't do the same thing. That neighbor communication is mediated by secreted peptides and a set of receptors. There's also more broad information, systemic signals about mediated by plant hormones or nutritional status, and even signals from outside of the plants, like these environmental cues that I've been hinting at for the last bit. So our question then is, how do you approach all of these things? How do all of these signals get together and eventually give you a plant that makes stomata, makes the right number, and makes them in the right place? And it turns out that when you look at many aspects of those signals, not surprisingly, when you've got something that can be modulated by the environment, the place that it gets modulated is at the flexible beginning of the pathway, and the beginning of the pathway that happens to be mediated by that transcription factor speechless. So what I'm going to talk about now is a little bit about our interests and our approaches to getting at this flexible development, this environmentally sensitive development. And you can really approach this in one of two ways. You can be very process focused and say, there's this amazing stage in plant development where cells have flexibility. They can divide in different ways. They can divide a different number of times. This is indicating that this is an event that can go on many different times. And depending on how they divide, they can create more stomata or more non-stomatal cells. So how is it that this process is controlled? The other possibility is you can take a very gene-focused approach. I told you speechless is a key regulator of this, so what controls speechless? Now, as an example of something that's a process-focused approach, I'll refer you to a paper that we published a few years ago by Anne Vetan. She was very interested in cytokinin. Cytokinin is a hormone that is involved in many different developmental events. Typically, we think of cytokinin as promoting cell divisions. Well, this is a cell division type event, but there's not just cell division, there's different types of cell division. And what Anne was actually able to find by studying this process, very carefully looking at what happened early on and then how this affected stomatal production later on, she was able to identify specific elements of the cytokinin signaling pathway that were present within stomata. And it turns out that all of this lovely system here also ends up being mediated through speechless. So with trying to study a process, we end up back with our gene. So let's go back to this gene-focused approach. It's an attractive approach to understanding development because we've got this molecular anchor. It's a transcription factor. We know what to do with transcription factors. They control other genes. They might have partners. And so we can use this as an anchor to figure out what other genes are involved in stomatal development. And perhaps if we understand what is upstream of speechless, we can get a handle of those environmental events. So just a few references related to this. So Andrea Mer, who's a postdoc in the lab right now, has really pioneered proximity labeling or very sensitive approaches to finding proteins that interact with very rare proteins, and so she's been able to build out the stomatal lineage that way. And Ansan Lau had studied speechless and in terms of understanding its targets genome-wide. So we've got anchors that we can look at in Arabidopsis, but another thing that's really nice about a gene-focused approach is that we can start to look at other plants. And as I mentioned before, these stomatal BHLHs have features that allow us to find them across many different plants. And so one of the things that we can do is leverage that conservation to explore the roles of the beginning of the stomatal lineage and speechless in diverse plants. And then we can also take advantage of work from other people. Dan Chitwood, when he was in Nilima Sinha's lab, and Chris Muir, were using the natural variation in tomato and other Solanaceae species. And actually in some of their studies where they were looking at leaf properties and studying stomata, realized that underlying a number of the QTL for some of these traits was actually speechless. And so we have some ideas that speechless might actually be underlying some traits of importance. So as a way of kind of illustrating how we might look at speechless regulation in terms of environmental effect, I'll just tell you a little story from Ansan Lau. This is work that he did actually when he was starting his own lab. And Ansan had actually studied a number of different environmental inputs into plant development. So he had worked on light signaling earlier and then was working on speechless and stomata. And one of the things he got interested in was temperature. So many plants change the number of stomata they make under different temperature conditions. So in Arabidopsis, if there's stomatal index refers to the ratio of stomata to other cells in the epidermis. And so there's a certain ratio under ambient happy Arabidopsis temperatures. If you grow plants for a few days at that temperature and then shift them to a lower temperature, they actually make more stomata. And when you switch them to a higher temperature, they make fewer. So he was intrigued by that phenomenon and was able to actually trace that to the differences in behavior of speechless. And to just show a little bit of data about that, this is a reporter that's reporting speechless transcription, not the protein, but the transcriptional reporter. And under ambient temperature of 22 degrees, there are many cells in the stomatal lineage that have high levels of speechless. But at high temperature, there are fewer cells and the levels are much lower. So something was controlling speechless. A good candidate to control speechless was something called PIF4. This has been worked on by other groups that were studying PIF4's regulation role in regulating temperature. And it turns out that this is the speechless gene right here in the promoter region, that PIF4 was able to bind to speechless, but only, in these pink bars, only at high temperature. So what was really exciting about this was it was a link between an environmental effect mediated through a transcription factor to an output of speechless, and then an output of cells that did or did not divide and did or did not make stomata. So seeing that that was the case, that there were ways that speechless might be regulated by environmental factors, this inspired Ido Neer, who's a new postdoc in the lab right now and was working on tomato. I mentioned that speechless is conserved across a number of plants, and so Ido started to look at the speechless locus. Now, if we remove speechless from tomato, we're not going to have any stomata at all, and that plant will die, so that's not so useful. But with new techniques that were pioneered by Zach Lippman, Ido took the approach of saying, let me essentially mutagenize by using CRISPR-Cas9 the promoter region of speechless in tomato. And so he made a number of small deletions all throughout the promoter region and asked, did any of those affect the behavior of the production of stomata or the behavior of speechless? Now he had some that eliminated a lot of this, and then we didn't get any stomata. That was a good control. But there were others that were small deletions that when he grew them in our greenhouses under normal conditions, they didn't look any different. What was exciting though, was that in one of these regions, a deletion of that region actually led, and I have to give a warning, this is preliminary, and with the current environment, it's going to be a while before we get to retest this, but it looks pretty good. And it turns out that he's got a version of this that seems to be insensitive to warmth. So normally tomato also change their stomatal index with higher temperature, and this one in this mutant is completely insensitive. Okay, so I gave you a couple of ideas about why we're excited about stomatal development, and particularly as a way of understanding how environmental inputs come into a developmental context. So it's obviously ask what's next. I think at this point, we know a lot about these core genes, these core regulators of stomatal development. And so how can we build from that? One thing we could do is build in more detail. So we have some core regulators. How can we understand the details of this stomatal development process or the behavior of these proteins? And some of the tools that we can use have to do with the imaging that we've developed and the ability to track lineages over time. So this is an example. This is a movie from graduate student Yang Gong. And so this is actually watching cells divide in the epidermis and watching another feature of these asymmetric divisions, that is that they're polarized. So that's kind of fun to watch. And so some tools like this have been very useful in understanding what's going on in development. Other tools, such as those developed by Camila Lopez-Anido, who's another postdoc in the lab, is to use single cell sequencing to actually explore variation within the stomatal lineage. So this is how we can go deeper. But as I've focused on in this talk, what our real interest is how do we go from this core up to the whole tissue, organ, plant, and ecosystem scale. So I want to make a shout out to the stomatal team. This is the team as of last year. I'll show you a couple pictures in a moment with some of the key features. This is Jan who did the imaging. It's Andrea. Where's Camilla? Camilla in the front, and Ido on tomato. But I also wanted to note again that this is our almost current team. But a number of the people that I mentioned, stomatal development is really happening all over the world. There are a couple of key places for stomatal physiology, Sheffield in the UK up here, ANU really in Australia is really key as well. Keiko Tori's at University of Texas now. But a number of the people that I mentioned here in pink are really, they're starting their own groups and really developing some wonderful stuff. So I like that global nature. And of course, our current lab actually looks like this. We do a lot of Zooming, and so I want to thank them again for their patience. And if I have some time for questions, I am happy to take questions. Thank you.
Thank you. So yeah, the questions are coming in. I think kind of touching on stomatal development, how it intersects with the environment and all.
I'm sorry, I'm having a very difficult time hearing your question. It's cutting out quite a lot.
Could you hear this, Dominique?
Okay, I can hear that, yes.
Okay, so the first question was around this sensing, the temperature sensing. So you showed that the question is before able to bind to the insensitive version of speechless in tomato. I guess we might have been not that far, but that was one of the questions.
So that is a really wonderful question that we would love to know the answer to. So one of the things that we did early on was to ask, could we just compare the promoter regions between speechless in tomato and speechless in Arabidopsis? They're very different. And so while the protein is quite conserved, it was difficult to say, let's find that same binding spot within the promoter of the tomato version. But of course, that's one of the things that we want to do with now that we have things that are maybe functionally insensitive to temperature. The prime candidate would be something like PIF4. Yeah. But we need to have a lab started up again to do those experiments.
Yeah, so there was another question about the temperature sensing. So did the person cut it right that in tomato, if you shift temperature upward, you actually get more stomata, while in Arabidopsis, you actually get less? Could you speculate, like, was this the case? And if so, could you speculate why this would be the case?
So very observant. I'm really happy that someone saw that. So I didn't actually mention it, hoping that someone would notice. So we've verified this. We've done this a couple of times. Tomato. So one thing is that tomato likes warmer temperatures than Arabidopsis. And so we had to change the temperature range that we did this in, but it was still consistent that when we raise the temperature in tomato, you get more stomata, and when you raise the temperature in Arabidopsis, you get fewer. So that's a really interesting question. Why do they do that? Is there something about, when Ansan first got the result in Arabidopsis that they made fewer stomata at higher temperature, I thought, that doesn't really make sense to me. I would think that at higher temperature, you'd want to transpire more and cool. So that was counterintuitive. And that's actually what tomato's doing. So perhaps for tomato, transpiration to cool is a really important thing. Now, maybe for Arabidopsis, higher temperature often is associated with drought. So maybe there's some trade-off. And that in some plants, it's better to not make as many stomata because high temperature is typically a water deficit, whereas other ones, it might still be a water deficit, but the chance of overheating is even worse. So we don't know. And I'm totally speculating, but I think it's really fascinating. And I think it's also fascinating that it's important to look at a number of different plants, because for sure they're all having environmental responses, but they're not always going in the same direction. And being able to also explore the development a little bit more. Why is it? We have a pretty good handle about what types of cell divisions lead to more or fewer stomata in Arabidopsis. We don't know that so much in other plants. And so as developmental biologists, we're really excited about that.
Yeah, it sounds really exciting. So a couple of questions were around into similar directions. So is there any like multiple alleles in Arabidopsis for speechless or other of those components which shape maybe the environmental response, and also in crops, and has this been selected for during breeding processes?
So it's a good question about the breeding processes. One of the things in grasses, so things that we eat a lot of, wheat and rice and maize, speechless has actually been duplicated. And although we don't know so much in the actual crop plants, in Brachypodium, which is a model that we use for grasses, it looks like speechless has kind of a dose-dependent role. So if you eliminate one of the copies of speechless, you get about 20% fewer stomata than you would if you had both copies. And the other one is a major influence. And if you get rid of both of them, then you don't make any stomata. So the way that it's going to play out in grasses is going to be quite different, but potentially quite interesting, because this is a situation where if you completely eliminate one, you still are alive and you still make stomata. So you might be able to see more differences there. Now in Arabidopsis, there are a few alleles, mostly having to do with phosphorylation sites. So people created various hyperactive versions of speechless. But no one's actually tried to do the promoter alleles like we've done in tomato, but it might actually be interesting to do that in Arabidopsis. It's mostly the Arabidopsis ones have told us a lot about the kinases that phosphorylate speechless, and those kinases also bring information. Some of them are MAP kinases, some of them are cyclin, cell cycle kinases, others are BIN2, so brassinosteroid related kinases. So often we've learned about signal transduction because of the speechless alleles more so than the environmental effects.
we have time for one last question. There are many more, and we're going to forward you them and put them on the Plantae webpage so you can pick and choose to answer them later on. And there might be also questions on Twitter later on. But the last one for today, right now, is coming to the MAP kinases a little bit. The question is, so does MAP kinase 12 affect the water use efficiency? And how does this affect the development of stomatas, either in density and aperture in response to drought stress?
Oh, that's a good question that I don't know all of the parts to. So MAP kinase 12, if I remember correctly, is one of the MAP kinases that is expressed, if not exclusively, at least mostly in guard cells. And a combination of that and I think MAP kinase 9 has some effects on stomatal behaviors. And there's a paper about it that I'm not remembering right now. So this might be a better one for the offline or later responses. I haven't read, and we haven't done any work ourselves on how the function of stomata might feed back on development. But that's an interesting kind of question. And it's an observation that if plants have mutations in which their stomata are not so responsive, so they're slow, or they're maybe physically not as good at opening and closing, then so those will be in the early leaves. And then what we see is as the new leaves emerge, the stomatal numbers, the density are often fairly different, as if the plant is trying to compensate for the lack of function of the stomata by changing the numbers. So that's an observation that I think could probably be an entire PhD worth of work if someone wanted to do that. But yeah, I don't know any more details about the MPK12 right now.
Cool. Thank you very much, Dominique. That was exciting and hands together. And thank you again. All right, so the next talk is by Matthew Gilliham. This is about GABA signaling in guard cells act as a stress memory to optimize plant water loss. So maybe getting a little bit to this MPK12 question. So Matthew Gilliham is director of the Reid Research Institute at the University of Adelaide. Matthew gained a PhD in ecology from Lancaster University and a PhD in plastic physiology from the University of Cambridge, UK, followed by a postdoc at Cambridge before immigrating to Australia, where we welcomed him. His research focuses on crop plant nutrition, stress resilience, with a focus on salinity and drought tolerance. Please take it away for us, Matthew.
Okay, thank you, Benjamin. Now, yep, hopefully everyone can see my screen. Good, excellent. Well, thank you, Benjamin, for the introduction, and I'd like to thank the organisers, ASPB, for organising this seminar series. I think it's a very important initiative, especially in this time of quite necessarily reduced travel or non-travel, and so hearing many speakers from around the world, it's a good opportunity for that. And also another shout out to ASPB, obviously being a society, they support a number of initiatives. They've got a number of journals, Plant Cell, Plant Physiology, and also an association with others, Plant Direct. And it's very important to support society journals. There's a special issue on transport and signalling. So if you're interested in submitting, please check out the website there. So today I'd like to give a talk about something we've been working on for a number of years now, which is really a new area, and I'm doing something a bit risque, talking about a lot of unpublished data. But this is centered around the molecule GABA, gamma aminobutyric acid, which many of you will probably best know as an inhibitory neurotransmitter. So it calms down glutamate activated action potentials. So I can't really talk to the veracity of the GABA being a health remedy in terms of a stress relief for mammals, but certainly for plants, what I'm hoping to be able to convince you of today is that GABA is in fact a relief for plant stress. This is the team, so led by Bo Zhu, who's a senior postdoc in the lab. Dr. Yulong recently moved on to an independent position in Henan University. And we have a group of very talented PhD students that challenge us all with some very novel data that we scratch our heads about. It's a very interesting project, and we're finding out a lot of new things. So I'm very excited to work with this team. It's not only a project involving us, but we've got collaborators around the world. I'd particularly like to point out Rainer Hedrick, who's a co-investigator on one of the grants that funds the most of this work. So a bit of background, gamma-aminobutyric acid, or GABA, rapidly accumulates in plants under stress, and it can increase in concentration by 100-fold in anoxia, and at least 10-fold with salt and drought and the like. It can increase quite rapidly. Even an insect crawling across a leaf can elicit increase in GABA concentration in the leaf. This, coupled to the fact that it's an inhibitory neurotransmitter in mammals, has led many to speculate over a number of years, over 20 years or so, that GABA is actually a signal in plants and not just a stress-induced metabolite. However, only over the last 5 years or so, we actually found a mechanism by which GABA could actually be transduced as a signal in plants. And that was a finding a few years ago in collaboration with Steve Tyman, also based in Adelaide. A bit of a shout out to Sunita Ramesh, that has also recently moved on to an independent position at Flinders University in Australia. We published this paper a few years ago in Nature Communications that shows an analogous system in both animals and in plants, where, and this is taken from a commentary by Victor Zarski, showing that obviously in animals, chloride concentration is quite high on the outside of cells. GABA binds to GABA-A receptors and allows the passage of chloride from the outside to the inside of the cell. In plants, what happens, because the gradient for chloride is opposite, so there's a high concentration of chloride in the cell, there are a number of anion transporters in the plant cell membranes that allow the passage of this chloride from the inside to the outside, and this depolarizes the membrane potential. GABA appears to be inhibiting a number of these anion transporters, so this essentially hyperpolarizes the membrane potential, keeping it in, say, a resting state. Recently, we found that the site that appears to be inhibiting the activity of these channels is actually on the inside. I won't go on into this in any detail, but this is also in collaboration with Steve Tyman. And so please look up this reference that shows that GABA is the acting from the cytosol. So what are these channels that interact with GABA in plants? They're known as ALMTs, aluminium activated malate transporters. It's a bit of a misnomer. They're named after the first member identified, that was from wheat roots, and this interacts with aluminium to stimulate malate efflux from the wheat root apex that collates that aluminium, preventing toxicity. There are 14 of these things in Arabidopsis, and only one of them is activated by aluminium, and most of them transport other anions like nitrate, chloride, and sulfate. They're known to be involved in a whole range of roles, from nutrition to pollen tube growth. And what we've shown is that, obviously, and many others before, that GABA increases during stress, but we've really shown that GABA interacts with these channels in a way. We've used a pharmacological agent to inhibit a physiological response. What we haven't done is shown that GABA has an endogenous physiological role. And so that's really what I want to talk to you about today. And the reason that I'm talking about Stomata, and that was a fantastic introduction from Dominique, So I won't go into this in any detail, other than obviously stomata are extremely important in terms of agricultural productivity. And in Australia, where drought is a key limiter to agricultural productivity, they're a key cell to study. So we're looking at the role of ALMTs in stomatal opening and closure. Stomatal opening and closure are based on the movement of ions and water, either moving into cells and increasing turgor, so increasing the aperture of this the stomatal pore, or the efflux of anions and cations and water, and the reduction of the stomatal pore. And as I just mentioned before, a number of these ion channels are actually these ALMTs. So basically, we're asking the question of whether, and this is a schematic of what's known about guard cell signaling a year or so ago. Obviously, I don't need you to take home all of this. It's built on the shoulders of giants, if you like, in the field. All we're really asking here is whether these ALMTs that sit in the guard cell interact with GABA and produce a physiological response. So the system we first chose to study this with was the standard assay of epidermal strips, so isolating the epidermis away from the mesophyll, floating it on a buffer, applying our agonist, GABA in this circumstance, and measuring the stomatal aperture. This is just to orientate you. So an open stomata in Arabidopsis will be about 3 microns, a closed stomata about a micron or so in aperture, and you can open with light, you can close with dark. So this is the initial assay that we performed. So in the constant light, we incubated the strips for an hour, either applied our GABA or not, and then waited another 2 hours and measured the aperture. And so you can see that the aperture here is about 3 microns. Without GABA present, when we add GABA, we get absolutely no effect. The apertures are the same. In the constant dark, we apply GABA a third of the way through. You can see that the stomatal apertures are a lot smaller here, nearer the micron. When they're in the darks, they're closed. We apply GABA, it doesn't cause a further closure or an opening. So that would be quite a disappointing talk if I ended there. GABA clearly isn't changing stomatal aperture. And luckily, we did another assay where we found that if we transition the stomata from the dark to the light with the presence or absence of GABA, this is the closed, this is the open stomata. So going from closed to open, you can see the apertures are increasing. In the presence of GABA, they don't open as much. So about an inhibition of about a third. You can do the same experiment going from the light to the dark, and we get a similar response. Again, GABA is inhibiting the movement. So GABA, in a way, is acting as a brake on movement. We've done this in a number of crops, so it's not only Arabidopsis, but broadbean, tobacco, soybean, barley. Barley is quite interesting, has a different stomatal architecture, but GABA is still inhibiting movement. And we've done it in the face of a variety of signals, so abscisic acid, carotene, hydrogen peroxide, amongst others, light and dark. And this worried us slightly because if GABA is inhibiting the movement to some degree, we might be permanently damaging them in some way, but we can wash out the effect. So after removing GABA, the stomata still respond fully to either opening or closing signals. Also, I guess it's important to mention, and please remember this for later on because it's an important point, GABA appears to be fine-tuning the guard cell movement. So it's not a sledgehammer effect, it's not a binary kind of effect. So low concentrations of low concentrations of abscisic acid, so that's obviously the drought hormone that increases during drought, close the stomata. When we apply GABA, again, it inhibits stomatal movement in the face of ABA. When we apply higher concentrations of ABA, the stomata close fully, even in the presence of GABA. So that's important to remember. I'll talk about that in later slides. The next point is we've got an analog of GABA called muscimol. It's a very potent analog of GABA. It's used as a diagnostic for GABA receptors in animals, and it seems to have a similar effect on LMTs. We can apply muscimol. It has no effect on aperture under constant dark or constant light. When we apply it during a stomatal transition from the dark to the light, we inhibit opening. And this is the important data. This is a stomatal conductance measurement, so using an infrared gas analyzer to measure water vapor, so the transpiration, if you like, from the leaf, going from the dark to the light. You can see that muscimol, and we've done this with GABA as well, delays and inhibits the rate of opening, and I'll show you some data later that it also reaches a lower steady state as well. So both GABA and muscimol do that. So it slows opening. So GABA appears to be a novel stomatal behavior modifier. It doesn't itself elicit a change in guard cell aperture, but it modulates also gas exchange. So what I've really shown now, again, is that it's a pharmacological agent that we can apply to inhibit stomatal movement, but does it really have an endogenous physiological role? The system we chose to study this was in the drought context. So we know that GABA increases in concentration in leaves, and this is Arabidopsis again. You can see that it almost doubles in concentration over a drought period. And this is from wild-type plants. And there are a number of genes that make proteins, these glutamate decarboxylases, that convert glutamate to GABA, and these are the major sources of GABA production in plants. The major gene conferring that protein, GAB2, is the major isoform in the leaf, and this is work of Frank Ludwig's a few years ago now. We took knockouts, a couple of knockouts of GAB2. They have no phenotype, visible phenotype, I should say, under standard conditions, so they grow the same size and have no visible phenotype, lacking GAB2. But if you do some biochemistry, you look at the GABA levels, they're near zero in the GAB2 lines. This appears to be the major isoform making GABA in the leaf, both under standard conditions and also under these drought conditions. So under wild type, it increases in concentration. Under drought conditions, there's no increase. So this GAB2 isoform appears to be the major isoform making GABA in the leaf, both under standard and drought conditions. So this is an interesting slide and a really key piece of data. What we see here is that when we remove GABA from the leaves in these GAB2 mutants, the stomatal apertures are much greater than wild type. So you can see here that clearly the aperture is greater. We did stomatal density measures, and there's no difference in density. So when you get a greater aperture and a similar density, what this has led to, again using an infrared gas analyzer, we get an increase in the stomatal conductance, so the amount of water vapor leaving the leaf in these GAB2 knockout lines. And this leads to, under a drought period, so we remove watering from the plants, compared to wild-type plants here in white, they have a lot lower relative water content, so they wilt a lot more quickly than the wild-type plants. You can see our knockouts here, they're wilting much more quickly than our wild-type plants. GAB2 is expressed everywhere in the leaf, but I won't be able to show you all the data due to time constraints here, but we focused really on the role of GABA in the guard cell. So it is expressed GAB2 in the guard cell, but it's also expressed elsewhere in the leaf. So we've expressed using GC1, which is a very faithful guard cell promoter discovered by Julian Schroeder's lab a few years ago now. Drives the expression of genes to a high level in the guard cell. We express now GAB2 in the guard cell using its promoter. And we expressed it in the GAB2 knockout lines. And what you can see here is it increases GABA concentration in those leaves. And the important piece of data here is that it restores the wild-type-like stomatal conductance of the GAB2 knockout. So it reduces the stomatal conductance, the water loss from those leaves. And hopefully it makes sense that we're losing less water from these GAB2 expressing leaves, specifically in the guard cell. And this leads to this wild type like stomatal phenotype during drought and water loss. So we get more water loss and more wilting in the GAB2 plants, but a lot less in our complemented plants. So guard cell specific expression of GAB2 complements the GAB2 knockout. You can see after day 8 here, this is just a visual representation of these plants. They're wilted in the GAB2. They're clearly not wilted. They're like wild type in our complemented plants. So guard cell specific expression of GAB2 is important. We've expressed GAB2 in other cell types without the guard cell, so specifically in the mesophyll, and we don't get this effect at all. So guard cell specific expression is to be very important. We can also express GAB2 specifically in the guard cells in wild type plants. And this increases GABA concentration in the leaf, reduces the stomatal conductance of these plants now expressing or overexpressing GAB2, and it improves the drought tolerance, if you like. So they wilt less after a 10-day drought period than wild type plants. So those further bits of data, hopefully you're convinced that guard cell specific expression is important in modulating plant water use, and GABA improves drought tolerance. What many of you might be actually asking now is, you've shown that GABA is likely to be a signal that increases during drought and it inhibits stomatal closure. How on earth can it improve drought tolerance if it's inhibiting closure? Well, that's because there's two sides to the story. One is obviously closure, but the other is opening. So if you open less, you're going to be reducing your water loss from the leaf. And we believe, in fact, and we've got the data to show, that a major target of GABA is actually LMT9. The closure inhibition is actually a lot less important in this drought response. So one piece of data in this is we know that when ABA concentrations are high enough, they close stomata. GABA has no effect on this. The other important, and I guess what we're seeing is that ABA can be thought of like a stopcock, preventing water loss from the leaf. ABA is more like a faucet, fine-tuning that water loss. Another important piece of data here is, again, stomatal conductance. This is our GAB2 line that lacks GABA. You can see quite clearly that the stomatal conductance, so the water loss from these leaves, is greater in these leaves that lack GABA. But when we open, so we go from the dark to the light, the rate of opening is also a lot quicker when we have no GABA in the system. And you can see that in the integrated measure here, the rate of opening is a lot quicker. The flip side of this is when we have muscimol or GABA, we reduce the rate of opening. They open less and to a lower level. And this is similar to what we see when we have the LMT9 knocked out in the leaf. They reduce their rate of opening, they open more slowly, and they open to a lesser degree than wild type plants. Again, this is the water vapor leaving the leaf. And this was published a few years ago by Alexis De Angeli, whilst he was a postdoc in Enrico Martinoia's lab. He's now got his own independent lab in Montpellier in France. So we believe that LMT9 is the major target here, and GABA acts on LMT9 to reduce the extent of anions moving into the vacuole, which helps after cations and water go into the vacuole, blowing it up, and this increases the stomatal aperture. So we're regulating the activity of this transporter. The stomatal phenotype of LMT9 knockouts is kind of similar to our high GABA phenotype, but they're more drought tolerant. So what we can see here, and this is the last piece of data that I'll show, or the penultimate piece of data that I'll show. This is our standard assay in stomatal apertures. when we go from the dark to the light, so we're opening, GABA has no effect. LMT9 knockouts are insensitive. So we remove our, if you like, GABA receptor. These plants are no longer responsive in terms of opening to GABA. And this is the last piece of data. This is using a cross of GAD2, which has this high stomatal conductance without LMT9. So when we cross LMT9 with GAD2, we complement the wild type phenotype. So we bring it back to wild type levels. So again, remove our receptor and it reduces stomatal conductance despite the GABA levels being low. So I'll just walk you through that data in the last couple of slides. So under wild type conditions, we believe that GABA modulates, acts as a brake on LMT9. So fine tuning the vacuole contents and stomatal opening. When we remove the brakes, when we remove GABA from the system, the vacuole can blow up to a greater degree, and the GAD2 phenotype of this large stomatal conductance and large stomatal aperture here. Under drought, what we can see is that GABA levels build up to a high degree and inhibit opening. So what we think of GABA here is, in a way, it's like a short-term memory. So GABA doesn't affect closure because the night period is so long, so eventually the stomata will close, but the next subsequent day, after GABA concentrations have built up, then GABA is inhibiting the extent of opening, so the plant is able to reduce its water loss. So this is the last slides. I'll just go through. Hopefully I've been able to convince you today that GABA is a novel signal that fine-tunes gas exchange and intrinsic water use efficiency. GABA regulates the rate of opening. LMT9 is a target for GABA. GABA interaction with LMT9 acts as a short-term memory of stress. And currently what we're doing is investigating the physiological role of these other LMTs in the guard cell to see what role they have. So I'll leave it there. I'd like to thank you all for your attention, and I'm happy to take any questions.
Thank you, Matthew. Very nice talk and very nice complementing Dominique's talk at the beginning. So I just posted a couple more information about the seminar series and how you can support our seminar series in the chat. In the meantime, a couple of questions for you, Matthew. So one question, there have a couple of questions around, does GAD1 or GABA has any roles in roots or during and other stresses like biotic stress response and so on? Do you know if it's only specific for guard cells, or does it also, is it more like another hormone?
Yeah, so that's a good question. So absolutely, there's a major isoform in the roots, GAD1, which I think the question alluded to. We're investigating what phenotypes those plants have when you knock out GABA in the roots. And yeah, we believe, in fact, there's been a few studies showing that that it might have a role in salt tolerance over the years, so GABA. And we believe that it somehow, our data is consistent with that. Again, that's unpublished, but absolutely GABA is going to have other roles in other cell types. Pollen tubes are another example. Some work with Jose Feijó over the last couple of years has shown that GABA could well be a signal. In fact, there was a paper quite a few years ago in Cell that indicated that GABA could be a signal there, and work of Jose's has shown that that may well be the case. So yeah, absolutely GABA is having a role in other cell types. We chose guard cells to study this because obviously signaling there is so well defined, and we were kind of amazed to see that GABA is in fact another signal on top. So and it's an easy cell system to study.
Yeah, so just going on from this, there was a question of around what is the effect of GABA and stomatal movement in ABA signaling mutants?
Yep, that's an excellent question. So, yeah, the interaction between GABA and the hormones. I mean, you kind of said, is GABA acting like a hormone? It's dangerous territory to speculate that it is a new hormone, but it certainly has characteristics shared. And yeah, I think the interaction with other signals is important, as I've said, and I showed. If that ABA concentration is high enough, then GABA has no effect. So, as I say, I think GABA is more of a fine-tuned control rather than the ABA kind of stopcock effect on transpiration.
Right, so the last question is about, we can take, sorry, there are many more. We again will post them on Plantae, and maybe Matthew can pick some of those and answer them later if he has time. So the last question for now was, GABA was reported to modulate virulence in certain pathogens, for example, Pseudomonas, and also know that several fungal pathogens produce GABA. Do you know what happens with pathogen-induced stomatal closure and how this might intersect with plant immune responses?
Yeah, again, a really good question. So I didn't show the data, but we have shown that it certainly interacts with flagellin or carotenosin signals. So beyond that, I can't say what its role is, but it's certainly, as the questioner indicated, GABA does increase during those kind of stresses. So I would not be surprised if it does not have a role, because we know that GABA does have an effect against those elisters. So absolutely, I think it probably has a role. But I think it's careful. We need to be careful to consider that GABA, as well as likely to being this endogenous signal, will have other effects on the pathogens. For instance, if insects eat high concentrations of GABA in the tissues, it has an effect on their physiology. And we know that GABA has an effect on quorum signaling and the like in bacteria. So there's likely to be several effects there, but it's not uncommon for a signal to have multiple effects. So yeah, I think it's an interesting area to explore further.
Cool, thank you very much, Matthew. Let's thank both Dominique and Matthew for two very exciting talks about something very, very plant science, such as Stomata, and they were very complementary, and I think they will be also really useful for students who want to learn more about plant and plant signaling. So before we close up, thanks again also for everybody who, for your patience, for not being able to answer, that we couldn't get to all of your questions. Also for people who signed up for talks, please be patient. We're really trying to develop a strategy to accommodate more speakers or in the future. And if you want to develop this vision with us, please get in touch. And also thanks to ASPP for making this happening. So I hand it back for Katie to close it all off. And don't forget, there will be another talk next Wednesday, and she will let us more, which will be again more into European time zone. Katie.
Thank you, Benjamin, for moderating, and thank you to both of our speakers for joining us today. Don't forget to join ASPB. We have a discount code, presents10, to receive 10% off membership. I'd also like to give another shout out to this plant physiology focus issue that's coming up. You'll see a familiar name on here. Matthew is one of our editors for plant physiology. So if you have any questions about this focus issue or really plant physiology in general, please reach out to us. And I'd also like to just mention that in addition to this seminar series, this webinar series, there are a lot of other online events that are happening around the world right now. We're working towards getting a global calendar together to accumulate all of those in one place. So be sure to check that out. And if you know of other events, there's a section on there to submit events of your own. And with that, thanks for attending. Here's some last minute details. So a recording of this webinar will be posted within the next few days, and you'll be able to find that on our website. And you can also follow us. We're also looking for sponsors. So if you're interested in sponsoring this series or donating, let us know at community@plantae.org. All right. Goodbye, everyone. Hope you have a great rest of your day or rest of your evening, and we'll see you next Wednesday.